People change.
Thanks,
Perfect timing.
Yeah.
We're all doing
It's a busy day.
Thank you, Adam.
Yeah. You're starting.
I'm right on up.
Okay. Sounds good. Great.
It's always fun. I've always had stuff. Kind of like these licenses out.
Good afternoon. It's 3:30. Perhaps we should get started on time. I usually am with the Zoom calls. You have to give it a few minutes, but I think we've got a good quorum and we've got a great afternoon. So once again, thank you for coming to the launch, the official launch of the Bloomberg Schools Innovation Translation Council. We greatly appreciate you participating today, as well as we greatly value your partnership moving forward. A little more than a year ago, before the current funding climate makes portfolio diversification seem very imperative, I was asked to phase out the school's historical tech transfer committee. This was a committee that had been around to try to spur some innovation translation activities, but with the increasing role of Johns Hopkins Tech Ventures as the university leader in this area, this committee had been disbanded. What might have seemed like a routine administrative task, however, turned into something more compelling. Probably more compelling than Ellen ever anticipated as well. Naturally curious, I dug more into gathering data, engaging with numerous internal and external stakeholders, and ultimately developing a white paper that summarized recommendations for strengthening the school's innovation and commercialization activities. And really four themes emerged from that effort. The first is that attitudes towards partnering with industry have shifted. When I joined the faculty 20 years ago, there was a strong reticence to working with commercial partners, and that has changed, and now there's substantial enthusiasm. Second, the school has a performance gap in this space. Despite an annual research portfolio measured in the hundreds of millions, outside of one long-standing commercial winner from 30 years ago, our annual commercial and licensing royalty revenue is measured in the tens of thousands. To me, this underperformance indicates untapped potential. Third, faculty needs support. Many researchers don't even consider commercialization. It's just not inherently at the top of their mind. And when they do have an idea that they think might have commercial potential, they have so many competing interests with limited bandwidth to really pursue it effectively. Okay. Finally, this isn't just about revenue. As you think about the things that we do in this school, and a resounding theme around our school is the emphasis on broader global and public health impact, not just on $ return on investment. Innovation translation can be a tool for mission-driven impact. It allows us to translate public health solutions developed through rigorous science into the world where they can make a tangible difference. Following this work, Dean McKenzie and I had a series of conversations to shape the vision of what has become the Innovation Translation Council. Ellen also readily identified Professor Anthony Leung, who you'll be hearing from shortly, as the right person to lead this initiative, and it's already bearing fruit. Thank you, Anthony. As many of you know, Dean McKenzie is nearing the end of her remarkable tenure as Dean, and I want to take a moment to thank her, not just for her leadership over the past 8 years as dean, but also for her receptivity, her vision, and her continued support to the Innovation Translation Council as we launch it here today. So with those brief comments, please join me in welcoming Dean Ellen McKenzie.
Well, thanks so much, Greg, for those kind remarks. And we were determined to get this over the finish. Well, Greg was very determined to get this over the finish line before I exit out of my dean's role. And I'm just thrilled that we're at that point. As Greg mentioned, our school has long recognized the growing potential for innovation and commercialization to extend the reach of what we do in public health. These opportunities, as he rightly pointed out, it's important for you to understand that these opportunities did not begin with the recent changes in the funding landscape, although there are a lot of faculty who are a little bit more receptive and eager to jump in. But they have become more vital than before. The Innovation Translation Council was created, and this is important. It was not created in response to scarcity, but in pursuit of impact. And we keep coming back to that mantra. We're getting into this because we want to have broader impact. It reflects our deep commitment to ensuring that the discoveries made here by our faculty, our students, our collaborators don't end at a publication, but move into the world where we can do the most good in helping people achieve health. Public health has always been about bridging science and society, and the ITC is a natural extension of that mission, helping to translate research into real-world solutions, digital solutions and apps, data algorithms, services, technologies, and therapeutics that can save lives at scale. We recognize that not every challenge can be met through traditional government or nonprofit pathways. We're learning that just about every day. Sometimes market-based strategies can accelerate access, sustainability, and reach, especially when guided by our values and driven by the needs of the communities we do serve. With the launch of this initiative, we're doubling down on what makes our school unique, a relentless focus on health equity, scientific excellence, and very importantly, social good. The ITC will help us unlock new routes of discovery, collaboration, and importantly, impact, amplifying our work far beyond what any one grant or paper could ever achieve. And as Greg mentioned, we're especially pleased that Dr. Anthony Leung, who's a professor in biochemistry and molecular biology here at the School of Public Health, agreed to take on the role as director of this initiative, bringing together his world-class science and hands-on commercialization experience. But Anthony is not doing this alone. He is doing it in partnership with Greg, and Greg has been a guiding force working with Anthony to get us to this point, but also Anthony has assembled a great team, which he'll talk a little bit about in a few minutes. Anthony approaches this work with a collaborative entrepreneurial spirit that has already jumped in with great vigor and excitement. We're so fortunate to have his leadership as we launch this important effort. And with that, I would like to ask Anthony to come and introduce our speaker for today. Thank you.
It's an honor to introduce someone who has shaped the world of science and had a lasting impact on me personally, Dr. Phillip A. Institute Professor, recently emeritus at MIT. Dr. Sharp is best known for his Nobel Prize-winning discovery of speed genes, a breakthrough that fundamentally changed our understanding of molecular biology. But his contribution didn't stop at the lab bench. He went on to co-found Biogen, one of the first biotech companies, helping to launch an entire industry that continue to save and improve lives around the world. A fitting call to our motto, millions, saving lives, millions at a time. When I asked him during a call last summer why he decided to start a company, his response, and I'm paraphrasing, was, "It is about benefiting our children's. There's no better legacy." That answer captures so much who he is, deeply committed to fundamental science discovery, but also translating this discovery into impact. His accolades are many, from election to the National Academy of Science to the receiving the National Medal of Science. And if I list them all, I will use up all his keynote time. But beyond the honors, what have stayed with me is his generosity as a mentor, his boundless curiosity, and his belief that science must serve society. I have the great fortune of working with Phil as a postdoc at MIT. Even now, we talk several times a year. He will send me a new paper and say, "Let's discuss this." We were just talking about P53 earlier on, tumor suppressor genes. His enthusiasm for discovery is infectious. He also introduced me to commercialization opportunities in my career. These insights that have shaped how I think, how I lead, and how I really try to mentor others in return. So today, as we officially launch the Innovation Translation Council, it feels especially meaningful to welcome someone who has lived the path from science to innovation to impact. Please join me in welcoming our keynote speaker, Dr. Phillip A.
I don't think you should ever have a postdoc fellow introduce yourself before. Okay, thank you. So it's an honor to be here and to particularly participate in this exciting launch. I come from MIT, and innovation at MIT is part of the water you drink there. It's an essential part of the institution's history. I'm going to talk about innovation, which is different than discovery, different than patenting. Innovation means you have impacted on others with your work. I'm going to talk about biotech as an example of how that emerged in this need for innovation. And then I'm going to talk a little bit at the end about convergence, which will touch some aspects of AI in public health, but only tangential as examples. So I have some conflicts. The only one relevant to this is Adilum, and I'm going to talk about their work in part of the presentation. I came to MIT in 1974, just at the moment that mankind had discovered the technology to do synthetic genes, synthetic biology. With the development of recombinant DNA, how to join DNA together, how to sequence DNA to know that you have the right combination of segments to make what you want, and how to chemically synthesize DNA. All of this happened in the mid-70s, and it gave rise to an opportunity to found a whole new industry, and I would call that synthetic biology. And it came to me in a context in 1977, same year I made the discovery of split genes, as was described, by a call from a venture capitalist who was an alum of MIT, and invited me to fly to San Francisco to consult on an investment in the first biotechnology company, Genentech. So I checked him out, Ray Schaefer flew out, and they made the investment. And we started talking about this whole issue of what recombinant DNA and innovation would mean in society. And that led to the founding of Biogen in 1978. And this is Wally Gilbert over to the left and myself peeking around the corner. And Charles Weissman here and Ken Murray and Bernard Mock. And we met, and the company was founded in Geneva, Switzerland. And we met there several times a year with the objective of creating a private sector investment professional community doing recombinant DNA. Because we felt it was going to be a very important activity in both pharmaceuticals, energy, chemistry, biological systems. We're going to take on this synthetic approach to impacting on society. And I was trained as a chemist at the University of Illinois, and I had learned about how synthetic chemistry had changed a whole industry in the country and the world. In 1970s, this is what Kendall Square next to MIT looked like. You see to the right of what is Main Street here, buildings from MIT. To the left, empty parking lot. And Biogen started with its first laboratory over to the left there. This is New England. There was a recession in 1978, the early 80s. And now what has happened in that space is that this is the same view. The building there is the Draper Lab. MIT is to the right. And all of that building in the midst of all of that is biotech, founded around this whole issue of doing synthetic biology, recombinant DNA to impact on society. Biogen owns, and at that stage, occupied about 3 buildings in that cluster. Now, MIT is not the only important issue in this whole development of biotech in Boston and Cambridge. Right across that river is MGH, which has a magnificent faculty of doctors and physicians who not only treat patients, but understand disease processes. And right up beyond us to the north is Harvard. So they're all on a red line. They were all associated with MIT, and they've contributed enormously to the development of biotech in this region. Flash forward to 2008. This whole engagement in biotech and pharmaceutical development has turned into the Kendall Square cluster of research science. Not only is there about 50 or so biotech companies located in this area, some of which are 5 people, some of which are thousands, but in the area, large pharmaceutical research has established in the community. Novartis has its international research on Mass Ave, immediately upstream from the red buildings that are MIT, as you see on the Charles River. Sanofi bought Genzyme and moved into the community. Pfizer has 2 buildings right north of the campus. Biogen has 4 buildings there. And I want to particularly call out Alnylam, which I'll talk about in a moment, as a biotech development, and the Broad Institute, which was involved in the Human Genome Initiative and brought big data and high throughput and high complexity biology to this whole endeavor, accelerating greatly the development of biotech. The Broad Institute was established by Eric Lander, who at Whitehead started this initiative in human genome sequencing and human genome genetics in the early 1990s, and then created, after the completion of this genome sequence in 2003, the Broad Institute with the philanthropic support of Eli Broad. And that then became a highly multidisciplinary research institute on the edge of MIT, committed to high throughput sequencing, the development of high content analysis of biological diseases and human genetics, and ultimately changed and impacted almost all of biomedical research. The most important aspect of this is to decrease the cost of sequencing so that one could then begin to develop the technology to analyze the disease process at the level of single cells in tissue in the disease process. And this is currently revolutionizing how we do our science and how we project this in terms of healthcare. Just as an example of what emerged from this type of information is shown here from a review article in 2015 about the genetic burden one finds in human genetics. And what is in 2015 about 3,000 Mendelian disease genes that are segregated by Mendelian inheritance is now close to 5,000 or 6,000. About 8% of all live births have a mutation or variation that impacts on the health of the individual throughout their lifetime. The estimated cost of this for a child born with a severe handicap is close to $5 million over their lifetime. And the diagnostic rate is about 11% for children, which show phenotype at the age of a small infant, and about a 30% for adults. But what this says is there's a very significant genetic burden that one can begin to analyze if one has the sequence of a genome of an individual. That sequence now costs less than $200 in a high throughput facility to produce a sequence capable of identifying a disease variation. So this developed all in 2000. At the same time, many scientific discoveries were also accelerated by this technology. And one of the more impactful ones, and as an example of something that we were involved in at MIT, is the discovery by Andy Farr and Craig Mello of RNA interference. Andy Farr at that stage was in the Carnegie Institute associated with Johns Hopkins here in Baltimore. He had been a graduate student at MIT and did his PhD work in my lab. He arrived at MIT at 18 years of age. He had a hairdo like this. He was a math major. He had had one course in biology, and he wanted to get his PhD in biology. And I interviewed him, and this person is really going to be able to do something. And he did a very good thesis and then went off to England with Sidney Brenner. Craig Mello was out at the University of Massachusetts. And what they discovered using the worm C. elegans was that if you made double-strand RNA from a given gene in C. elegans and fed it to a worm, Geraldine here at Hopkins did that work with Andy, you fed it to a worm, you would silence a gene. So here, double-strand RNA from a DNA sequence of a gene introduced into this worm silenced the gene. If that could be translated to higher order systems, mammalian systems, it would have an enormous impact. Tom Tuschl and Phil Zamor at MIT with David Bartel synthesized double-strand RNA and discovered that this process could occur in an extract in vitro using tissue from worms, or in this case, Drosophila. And the active component that was then elucidated by the work of Tom Tuschl is what we call a small interfering RNA, a 21 nucleotide RNA that is complementary to a gene, messenger RNA. And if that RNA enters the cell, it pairs with a messenger RNA and then silences that gene. Now, if you think about it, oral drugs silence genes by interacting with proteins like enzymes in the cytoplasm of cells. Antibodies silence genes by interacting with cell surface markers. Here we had a way to interact and silence a gene specifically at the stage of RNA, something that really had not been possible before in a very systematic way. Now, this got Andy Farr and Craig Mello a Nobel Prize in 2006. And the reason that this process works in our cells was the basis of a Nobel Prize to Victor Ambros and Gary Refkin this year in 2024. And what they discovered was that the whole process of genes in our cells, there was at least 500, this was in 2001, at least 500 genes that are encoded as microRNAs that then are transcribed by RNA polymerase, making this hairpin RNA that is processed into an siRNA type structure, and Dicer processes it to make a complex that silences a gene if it is exactly complementary to the small RNA, and silences a gene partially by degradation if it's only partially homologous. Our cells had a biosynthetic pathway where you could take small RNA and introduce it into the cell and silence a gene specifically. Here was an opportunity to both explore the biology of cells by silencing specific genes, as well as possibly being able to treat diseases with a small RNA, specifically to the sequence of a gene. And that led a number of us to co-found a second company called Alnylam in 2002. And I and our colleagues at MIT and Paul Schimmel was the scientific founders. John Maganori was CEO. John had worked previously at Biogen. I was on the board at that stage and chair of the scientific advisory board. I knew John well. We recruited him, established the company. In 2004, we did an IPO. And in 2024, we have about 3,000 employees. Now, this course of over 20 years cost about $2 billion. That money came from relationships with pharmaceutical companies, as well as relationships with public financing. And our first challenge was how to deliver these RNAs into cells. How do you get a charged double-strand RNA through the lipid membrane of a cell to be able then to silence the activity of a specific gene? And that led, if we could introduce it into the cell, entering this pathway, pairing to a gene in which we could identify the gene and the sequence of the gene from the human genome sequence, have it cleaved and silenced in a catalytic way. And to do that, we were able to develop a whole technology of lipid nanoparticles, which depends on the nature of the lipids that are part of this particle. And Peter Cullis in Canada showed that cationic lipids have this property of being able to assemble on nucleic acid, form a lipid membrane that would fuse with a cellular membrane. And when we took this technology and perfected it, what we found is that those lipid particles associate in the blood with a protein called ApoE that has receptors on liver cells. and it is internalized from a pH in the cytoplasm of about 7 to an endosome about 5. And that change in pH changed the conformation of these lipids. And that releases the RNA from the endosome into the cell. Now, that technology was perfected by Alnylam. And what you see here is a test of the efficacy of that technology in delivering an RNA to the liver of an animal, in this case, rat. And what you see is there's a logarithmic scale to the right. And if you design those PKs to be about 6.2, you have a 3 order of magnitude increase in efficiency of uptake of that RNA. So here we have the technology to deliver the RNAs to a specific cell. And that technology was what was used when others developed mRNA vaccines to vaccinate us against COVID. The same technology in a lipid nanoparticle gives rise to the vaccination for viral infections. The same technology is going to give rise and is giving rise to vaccination against diseases like cancer and others where you want to stimulate the immune system to a specific antigen. So here are examples of how technology developed in an academic world around basic research has been translated to impact on society in ways that provide cures to patients who haven't been curable or treatable before, and to prevent and help control infections of various types. But when you step back at this and you look in the long term, these advances in biology, from our understanding of how genes and cells and cell biology processes work, have gone on almost in parallel with revolutions in engineering that has changed how we can think about an impact on biological healthcare problems. Information technology, which is clearly related to our ability to handle big data, to process and predict, to do AlphaFold and predict protein structures, to do machine learning to process all the aspects of how to predict and support a given decision-making tree. Storage and processing of large data. Synthesis of composites at nanoscales, so you can make a nanoparticle. You can make a delivery system that has layers of particles of materials that dissociate and release at certain time periods. Microfabrication, high throughput quantification, the ability to sequence tissue at a level of a single cell in a tissue, creating a disease process. And at the end, control theory and technology to think about dynamics of systems. If you think about what we've been involved in mostly in biology or molecular biology over the last 20 to 30 years, it's all equilibrium technology. We describe systems at equilibrium. We have yet to develop the technology and understanding to describe systems that are dynamic, that are using flow of energy to create a cell that maintains itself from equilibrium. And that process is something that is going to explore new realms of technology. But the other side of this coin, in terms of healthcare and these types of technologies, is the increasing cost of healthcare in society as we develop new treatments and new expectations for how these processes are translated to a patient. Now, this increase in cost is not an increase that's primarily driven by the increase in proprietary drugs. It is primarily increase in other causes in society. And I think it raises the question in healthcare delivery in this country. And it raises the question, is it the time to re-engineer healthcare? And I think it is the time to re-engineer healthcare. And I think that engineering has got to be done in a way that preserves the impact of new insights and makes delivery to the patient more efficient and more effective. So let's talk about that for a minute, because I don't have answers to this, but I do think it's something public health should be contributing to. What do we have as convergent? Now, these slides have been made for interception into cancer because that's the world I have done my research in. I've been head of a cancer center for 7, 8 years, and then beyond that, in a cancer center all my career. In the interception of diseases, you want to predict the risk of future disease development. Because if you can predict the risk in the future, you can intervene and control and ultimately get better control and prevent. You want to be able to accurately and effectively early detect, because if you predict, you have to accurately detect and be able to present biomarkers for it. And you need to design, educate, and evaluate the implication of this technology to provide access to people who need access to the healthcare. So access is an important part of how to deliver this healthcare issue. Now I'll give you a few examples, just as examples from MIT that I am familiar with. This is the work of Sangeeta Bhatia, professor at MIT, building on work that was actually primarily initiated or championed here by Victor Belescu here in your cancer center, where you could detect circulating DNA in blood of cancer patients, and by analyzing the structure and sequence of that DNA and the chromatin structure, as well as RNA that appears in vesicles in the blood, you can get insights as to whether a cancer has been reduced to such a level that it no longer sheds detectable material, and even identify what type of cancer it is. And that whole technology is incredibly important in terms of being able to have high throughput assays that allow you to accurately predict and ultimately treat this disease. And what Sangeeta Bhatia showed this year last year in Science is that you can increase the level of that circulating DNA by both two technologies. One, delivering a lipid nanoparticle, liposome, that engages macrophage and suppresses their uptake of this DNA from the blood, as well as by using an antibody that would actually bind to that DNA and chromatin structure and protect it. And by doing so, they were able to increase the yield of this circulating material by 20-fold, and being able then to increase the accuracy and the amount of information one can receive from these blood biomarker assays to detect cancer. And if we want to really impact on cancer, the idea and necessity of treating minimal disease is really extraordinarily important. Another example out of MIT is the work of Regina Barzali, who has been using machine learning to process radiological images of women in breast cancer and to predict the risk of breast cancer from those radiological images in 3 to 4 years into the future from the image. And she's developed this technology and published on it extensively. What is shown here is a breast, I suspect it is her, that in 3 years progression, there was a cancer developed in this region that you can't detect cancer there by optical analysis, but by machine learning, they were able to detect it. So this technology seems to be, and is published as more sensitive than most radiological image analysis by physicians, but has to be done under a physician supervision, and allows prediction into the future. Now, that raises all sorts of issues about where you train the radiological images on and how you get a culturally diverse set of individuals and be accurate. So that over the last several years, about 5 years, has been implemented in various parts of the world. And the accuracy of this process is independent of the cultural origin of the patients in which it is being tested. So this is being implemented and tested in Mass General Hospital and other places, in Kaiser in the West Coast and others, and is a process that will allow more effective image analysis and radiological control of this type of disease. Now, out of that type of standardization becomes other questions, and one of which is shown here. Federal law requires mammograms to provide breast density notifications. Right now, these are a series of images of different breasts that she's provided, low density, mild density, higher density, and extreme density breasts. And under this law, about 40% of all patients are defined as dense by these types of analysis. And when you analyze that large segment, and this is work that is ongoing, the 40% of patients that are classified as dense for future monitoring as a pool have almost no increased risk relative to the total pool. So there is clearly a risk factor in some dense breasts, but it isn't sufficiently enriched in this part segment in her analysis that would warrant this type of notification and very tight monitoring screening, which raises the issue, is the healthcare system willing to use this type of technology if it is proven effective in a controlled study to actually focus on those subset of patients who really need to be monitored versus a larger segment of them who could be monitored but may not have an increased risk factor. So I see the opportunities for interception, or the equivalent in public health, of support and development of value-based evaluations of innovation in healthcare as to the patient benefit, well-being, productivity, and cost. That value system in application of translation of new technology to the patient, access to healthcare delivery, diagnosis consulting, supported by an informed staff. And by that I mean there needs to be greater engagement with the healthcare system with patients. In Boston, it is very difficult to get a primary care physician. It's a shortage throughout the country. We need to expand by technology access to healthcare through people who can engage with patients and provide the interpretation and support by our increasing amount of information we can bring to the interaction of the patient in terms of their health. That needs to be staffed by primary care physicians, and I think as well increased utilization of physician assistants and nurse practitioners to actually put person-to-person engagements in information for patients to digest. And supported by IT and information and machine learning type technology to be able to bring to that point of interactions between the healthcare system and the patient all the records, information of the previous healthcare system, and an analysis that would give an insightful future prediction of what this patient needed and how this patient needed to be encouraged for a given course of treatment. So I want to thank the people in my lab who worked over the years that I have been at MIT. I've not spoken a great deal about their particular work. And I also want to thank, Alnylam and its colleagues for taking these early embryonic observations and turn it into a therapeutic modality that's benefiting millions of people around the world. And I want to thank you for this opportunity to talk about the benefits of taking innovation and having an impact in the world, seeing it engaged in the benefit of patients, and trying to think about how to improve and advance healthcare in general in the country. Thank you for your.
I'll always start. Bill, on your early map of the Keynote Square area, there was a lot of orange that represented capital investments. And I just wanted to get your thoughts about co-location. A lot of the interaction between industry and funders and scientists, you would think now with Zoom and everything else, you wouldn't necessarily need to be co-located, but you really emphasize with those maps how you have academia, you have business, and you have investment. Any comments about the co-location? We would love to have a corridor here. There's been lots of discussion. We have people who are working on that, yet we're not there. And any thoughts?
When biotech started, all the technology essentially was in academic labs. And you would turn, and I was there and I made the pitch to pharmaceutical companies, and they basically said, this will die in a year. There is no way you can have a new startup produce a pharmaceutical company. There had been one in the previous 20 years that became significant. So the students were necessary to move into these organizations, postdocs, who then staffed them and became professionals and then went on and developed. So that was part of the reason for proximity. But now you see big pharma moving in with established research programs, and they're located within walking distance, 5 minutes walk from MIT. And we, with all the technology we have, technology transfer walks on 2 feet. It's two individuals talking. And that seeds interactions before they go to this meeting and that publication, and 5 years later it becomes interesting. And with a concentration, whereas Biogen as the first company in Cambridge has populated the rest of Cambridge. And these leaders interact with each other. And they take the responsibility for mentoring young people entering the field. So it's a community that builds as a team and captures the newest technology that comes along. RNAi, for example, and other technologies that have emerged over the years. And that's been important enough.
Thank you, Dr. Sharp. I'm curious as to the figure you gave about the $2 billion early on. Did you find that some of the funders wanted to be like a sole funder for part of that financial commitment, or that they wanted to actually go in and partner with other folks in the industry?
So Alnylam started with a venture investment of somewhere between 10 and 20 million. And then signed a first collaboration with Novartis. And that then followed by a relationship with Merck. And then we did an IPO in 2004 and captured another 20, 40 million dollars. And then we formed several other relationships and did a secondary, almost went under, and then formed a couple of other capital deals. So these are all by bankers, primarily, or pharmaceutical executives. These people are evaluating what this technology means to them, if it works, and how they wanted to learn about it and to be involved in it. And that then produced a base of innovation within the company to do novel lipid nanoparticle delivery, optimization of targeting, how to design a disease-specific treatment, how to prove it was safe, how to get metabolism of all those lipids, how to then go into patients and show that it was effective in patients. So all of that took an enormous amount of resources. Ultimately, established this technology, so when messenger RNA came along, you needed a lipid nanoparticle technology, it was there. And if you read the New York Times this weekend, lower right corner, there's a little baby, a year old, who's alive because that lipid nanoparticles was used to deliver gene editing to her liver and allow metabolism that was functional.
Great presentation. My question is, going back to the number that you presented, about $2 billion that it took for over 25 years for treating diseases like cancer. What are your thoughts on what would be required for rare genetic diseases, or even genetic diseases that affect people primarily in so-called LMIC countries, like, for example, sickle cell disease, right? I mean, there's been talk about that for decades now, but what would be required to bring something to fruition that is also approachable by people, in LMIC countries?
Thank you. Once this technology is established, it's available. I mean, this lipid nanoparticle technology is being used around the world. This example I mentioned in terms of this baby was using it to deliver an RNA editing to kids. I anticipate that over time, this type of technology, particularly for rare diseases, will be used as in a hospital delivery system by hospital-like transplant. And that there will be technology where you could do a sequence, identify a disease process, and either use gene therapy or nucleic acid delivery as a possible therapeutic in that setting. Now, we're not there, but we're getting close. And I think it'll happen. But this technology, when it's scaled, it's still somewhat expensive for third world. There's no question about that. And it'll take a sophisticated delivery system or physician to be able to use it. But I do think some aspects of it, like vaccines and others, will be something that can be used in a variety and broad setting. But those types of questions is something that an institution interested in public health and expert in this process is clearly going to be the leaders in making it happen.
Thank you, Professor Phillip.
Where you at? Oh, hi. Yes.
I was wondering if, do you think that we needed to rethink the way that we calculate the cost effectiveness analysis to incorporate better these innovative solutions? Because the way that we do this was recommended 30 years ago. And now we are facing the new environment about innovation. So you think that we needed to reconsider the way that we calculate this?
I think the answer to your question is yes. I think we are going to need to be able to put great emphasis on the ability to identify patients who, with a specific disease process, would benefit from a given technology that can be used in the setting in which the patient is there. And that process has to then be communicated, identified by direct interaction between the patient and a healthcare giver or provider. And engineering that flow of information and how it is then told and interpreted and guided for treatment is part of how to make a better healthcare system that I think is incredibly important.
Hi. To talk about how to get things implemented by using an example that I went through. I got diagnosed as having colon cancer, and I went to the doctors. They said, "Hey, we got a 4-week waiting period. You're in the waiting period for the colonoscopy or whatever it was." And I said, "Fine, great. Give me an ivermectin script while I'm waiting, because ivermectin stops the stuff. It doesn't kill it. It stops it. That way it doesn't metastasize all over the place." And I got ripped by about 3 different layers above that, and everybody thought it was a horrible idea. And I couldn't figure out why they didn't analyze what happens to people in their queue and ways of reducing risk to that. Well, luckily I didn't have it, but to somebody who did, that would have been 4 weeks of allowing this thing to move around. And the organization had no understanding of, hey, I've got these people in a queue. Am I hurting them by keeping them in the queue? And is there something I should be doing? I'm wondering why there isn't like a machine learning thing or a consulting firm like a McKinsey & Company or something that would go in, analyze the records, force them to do things. The only thing I see that does that is a class action lawsuit. It's really an inefficient way of doing it. And if we rely on that, it's going to be 200 years before we see the things that you're talking about getting pushed down to the hospital aisle.
Yep.
So how do we approach that? Is there something that I haven't seen that would do that, or is that?
Well, I think that's just part of the challenge of bringing this technology. But we'll always have to have evidence that the technology works, that it's safe, and it benefits the patient. It takes,
or doesn't harm.
And it doesn't harm.
Or doesn't harm, because sometimes
No, I won't settle with doesn't harm. I will settle that it has to be safe.
Yes.
And it has to show efficacy.
Yes, but we need to get to the right people to do it. And I see lots of stuff, but I don't see getting down to the right people.
Yeah.
Thank you.
Thank you.
They say between the idea and the reality lies the shadow. And 90% of companies succumb in the valley of death. You have succeeded in establishing very successful companies beyond vision, perseverance and ingenuity. What is the secret sauce to take it?
The secret sauce is to have really important new technology. You can't do a Me Too. It costs too much money to do a Me Too from a startup position. You have to have some discontinuity type technology. And even with that, there will be a day in which you will say, I'm not sure we're going to be here a year from now, all right? I need to make decisions to give me hard decisions, difficult decisions, frequently involving reducing staff and reducing your possibilities to a few targets, to basically be able to succeed in arriving to a point where you can basically feed yourself with resources from your business. The valley of death is always going to be there in a startup. It's the nature of the beast. You have vision, you have hopes, you have aspirations. If they're not based on conclusive facts. They're based on the insight that if this works in this disease or in this time, it will benefit patients. Can I get there? Can I do it? Can I show it? That requires grit.
Last question before we move to the panel session.
Well, is this working? Or does it have shout?
Just hold it straight towards your mouth.
Hello?
Yeah, it's a little bit.
Okay, well, I'll just shout.
Please.
I'm sorry, I'm not a scientist, so much of this went over my head, but I spent 30 years as a corporate lawyer, so I do have some interest in how you set people's incentives, both at the individual level, the scientist level, and the organizational level, to encourage the merging of private sector enthusiasm for wealth creation and the academic interest in, first of all, research and then impact on the world. And I think in my experience, I've seen it done well and done badly, but the one thing that I have often perceived is that there is not an institution-wide understanding and policy for how best to attract capital, but also how best to incentivize the academic staff to participate in a way which doesn't spoil relationships amongst academic staff, like somebody wins the lottery and other people don't, and in a way which incentivizes the institution to get behind this sort of thing and see it as in its self-interest to make it grow. Now, it's clearly worked at MIT, and I'm wondering, particularly given that this is of obvious interest to Johns Hopkins, I'm wondering if you have views on the secret sauce that MIT has obviously discovered as to how to get all the incentives right so that you get a bunch of bare land suddenly sprouting buildings with a whole lot of enthusiastic people doing great work and attracting the private sector as well as academics.
A question we discussed at lunch today. Thank you for the question. I will say MIT is a tech school. 50% of its faculty are engineers. They were involved in everything from ship design 100 years ago to the core of the computer, to machine learning, to quantum computing now, to fusion. So it's an institution that lives beyond its walls, its mind and hand. So you need to move your knowledge and impact into society as a core principle of MIT. So many of the cultural issues that you've described, which are real in many places, is sort of taken for granted at MIT. And the motivation that comes from that and the gratification that comes to that is seeing impact. It's changing things. And it is extraordinarily important, both personally to you and to society. So I think for any institution, building bridges to students can learn, emulate in some cases, and impact on society as a group more than you do today, and you do it all the time here at Hopkins even today. It'll bring benefits. Thank you.
Thank you.